participating as a participant or a speaker. We have a lot of excellent expertise today. And what we're doing, what this is, just in case you don't know where you are, is the annual Molecular and Cellular Oncology Program Retreat as part of the Cancer Center, one of the 4 or 5 core programs. And what we're doing with our retreat this year is trying to kind of workshop and brainstorm through how one might address not only serving the Colorado population, which is also called the catchment area for Colorado is the state, but serving our population in terms of cancer patients, as well as delivering healthcare equitably. And so if you are a researcher who studies basic molecular mechanisms, it's sometimes hard to frame your work or understand how to do this. And so we decided to put together a panel of speakers who are really awesome in all sorts of different areas to help you brainstorm this idea. And I'll show you in a couple slides why. Just also to mention who we are, Tintin and I are co-leaders of the Molecular and Cellular Oncology Program. And here's what we look like these days. This is the goal of the annual retreat today is represented here in the schedule. So we have today, with some pretty tight timelines, 2 talks from our keynote, Dr. Icefeld, as well as Adam Green, people who will talk about their own work that addresses health disparities in the oncology space. And then we have people with various expertise representing the Center for Personalized Medicine, that's Chris Ginu, the Cancer Center's Office of Community Outreach and Engagement, which is Evelyn Barayo, and then Sean Davis, who's Deputy Director of the new Center for Health AI. And then of course, the most important component is you. And so in contrast to what you might have seen on the schedule, instead of having lightning talks, we had so few people that really have a solid handle on health disparity research as a molecular mechanism type lab, that we asked for people to just come and throw out ideas that they might have and get some feedback in a series of breakout rooms at the end. And so I don't want to put Alexis on the spot, but she did volunteer to have some idea of what they're thinking about in their lab on behalf of Srinivas Ramakandran. So we thought we'd use her to kick off the ideas and then go into breakout rooms and just discuss your science and what you think you could do to really align your research to health disparities research. So the main goal of this retreat today is really just to brainstorm and discuss how one can approach health disparities research if you're really a molecular mechanism type lab. And let me see, yeah. So some of the questions here we'll come back to at the discussion, so I won't go into great detail, but we've started to look at issues of, is your model system representing a certain ethnic background, or what does your model system represent? If you're using cell lines, that's a real obvious question. How do you find out about cell line ancestry? What kind of databases are out there? And things about Colorado. So what populations are underserved here? What minority populations do we have? What are the most important exposures that we have that are different from other places? And then as part of a secondary goal, we've started to curate a bunch of resources that are maybe helpful for us to achieve these goals. So here I've just put a few of the papers that we've collected. A lot of these focus on newer kind of ancestry oriented studies that tell you about common cell lines. But we're also going to put anything that comes from the discussion today, useful websites, useful databases. We'll curate that all together and put it on our MCO website. And then, of course, there's always money involved. And so we wanted to point out these recently published announcements from the NIH that focus on both, that fund R01, R21, and R03. And here I've just highlighted that these are announcements that are intended to encourage basic researchers to take on mechanistic basis for health disparity research. So I think, yep, that's the end of my little intro. So with that, I just will stop screen sharing. And I wanted to introduce our keynote speaker, who's Dr. N. Catherine Icefeld, and I said I'm going to do a fairly short introduction, so forgive me if I don't cover relevant and many awards that you've won over the years. But Dr. Icefeld is a really great representative of what we're trying to accomplish here, and that is someone who has done really basic research and then also has focused on health disparity and outcome research. So she is an assistant professor at the Ohio State University. I realize it's supposed to have a "the" at the beginning from your CV. And then, yeah, I'm sorry, just a quick note to Ellen, to make sure that she can screen share. I think you need to enable screen sharing. But also Dr. Icefeld did her training in Germany, both MD residency and fellowship, and then came to the Ohio State University and did it again, apparently. And there she worked.
Always fun.
So you're very well trained. There she worked with Clara Bloomfield and is now director, I believe, of the Clara Bloomfield Center for Leukemia Outcomes Research. So with that, I think if you have a title slide and can share now, I think I'll just let you take it away.
Let me see. I think I should be able to.
And yeah, the title is Racial and Socioeconomic Survival Disparities in Adult AML Patients.
So do you now see the only my slide or also my notes?
You can see the notes as well.
And I have to change it around.
Change screen maybe.
Yeah, exactly. I'm like flipping the screens in here. See from where I'm putting those. I think I have to probably see around these great.
And in the worst case scenario, I think you could email. Oh, that looks good, actually. Yeah, if you just screen share, I think it'll be big.
This way?
Sorry. Yeah, that's perfect. Yeah.
Perfect. Ha. I just had to change the parts around in here. Okay, so thank you so much, Patricia, and thank you all for the introduction and inviting me. Super excited to be here today to talk about the racial disparities in AML. I'm really looking forward to the discussions after, and hopefully there will be lots of interesting ideas coming up. And I would actually like to start with my acknowledgements for all the contributions of so many people for the data that I'm showing today. This includes the members of the Clara Bloomfield Center that Patricia just mentioned, statisticians, cytogeneticists, faculty members, the Institute of Genomic Medicine, like one of my mentors, Elaine Mardis, with whom I do a lot of the large-scale sequencing studies, and also, of course, the patients and families that enroll themselves and get treated on protocols of the Alliance for Clinical Trials in Oncology, which is a large cooperative group such as ECOG or SWOG, where over the past 3 decades, patients have been treated on. And we are really fortunate to have not only the clinical and outcome data, but also tissue bank associated with that, where we leverage a lot of our studies from. So it is well recognized that Black Americans suffer from worse survival outcomes across a broad range of cancers. And this is also reflected here in the most recent American Cancer Society Cancer Facts report, specifically dedicated to cancers in African Americans. And with respect to AML, what I'm going to be talking about today, the first larger study that really addressed the differences with respect to molecular features and survival between Black and white patients was conducted back in 2004 on behalf of the CALGB by Michael Zacharias, where the authors for the first time pointed out a higher proportion of especially favorable risk cytogenetics in Black patients. However, despite these seemingly favorable cytogenetics, white patients had improved outcome compared to Black patients, and in particular compared to Black males. And this disparity has mainly been attributed to differences in treatments received, as well as various socioeconomic factors, of course, which could affect treatment compliance, or also access to tertiary care centers, which have, as you all know, like a disease-specific expertise, and they have the infrastructure to place patients on clinical trials. And all of this points towards the elephant in the room, which always is there and which we need to address, which is the structural racism that really ultimately impedes care. However, talking about what we can do, even as basic researchers, over the past decades, there have been remarkable strides in the molecular characterization of AML. Thus, for our team, the question about mutational differences between Black and white AML patients, both with respect to the gene mutation frequencies and the associated prognostic impact, were questions that absolutely needed to be addressed. And this really came up from more out of a basic research that I was conducting like roughly 2 years ago. So why would this be so important to know? Because of the tremendous clinical implications that our increased knowledge about AML biology, and especially associated gene mutations, has brought upon us. So much so that we are not only basing our disease classification on it, but also our treatment decisions. So this is a graphic that you're probably all familiar with, which is the ELN Classification of AML in its most recent version from 2017. And as you know, there are select gene mutations that found their way into the classifications. But now one has to understand what these recommendations are actually based on. So the ELN is an international expert panel that makes its recommendations based on repeated findings published in the literature, and specifically the basis on the largest and most well-published literature studies. And while the ELN is not thought to be primarily a prognostic classification, you can see that applying the ELN criteria, for example, to our alliance patients, it does a pretty decent job in separating patients based on their survival. And it does such a good job that guidelines, NCCN, and ELN really, for example, don't recommend patients who belong to the favorable group to get an allogenic transplant in first complete remission. So sounds all good, but here's a caveat. The fact that these recommendations are based on the findings of the large genomic studies have to raise the questions about the patients that were actually included into those studies. So it's very important that we all understand that the genetic and genomic knowledge we have about AML actually stems by and large from white patients, because as you can see here, basically all hallmark publications about AML over the past decade have only had very small proportions of Black patients, and as a matter of fact, also any other non-Caucasian ethnicities. And this is pretty concordant when we look at the cancer genomic studies in general, where the inclusion report lists like approximately 2% of cancer genomic studies being conducted on Black participants. And so this big discrepancy in knowledge bias that we have based on this sparks the initiation of the first study that we did in adult AML, where we first wanted to see whether even now, it was almost 20 years after Dr. Sakhar's first study, survival disparities for Black AML patients persist, and using both a population-based analysis on almost 25,000 patients from the SEER registry, and also in the setting of clinical trials using our data from 1,300 AML patients. Moreover, we wanted to see whether there are any differences in the molecular landscape of known AML-associated features, and whether any of these genetic factors that we know also affect the survival of Black patients. So in order to have a little bit more time to show some newer unpublished data, I will only focus on the major findings and key conclusions that we had from this initial study. So with respect to the survival, the answer was very, very clear yes. Both in the analysis of the SEER patients and of patients treated on frontline alliance protocols, the overall survival of Black patients was significantly shorter than the survival of white patients of the same age. Second, in the population-based analysis, which had the ability to assess the impact of select socioeconomic features, we found that, as depicted here in the forest plot, Black patients had a 27% higher risk of death compared to white patients, even after adjusting for factors like age, sex, measure of poverty, and also decade of diagnosis. The third conclusion and novel finding was that indeed, differences do exist in the mutation frequencies of several well-known AML-associated genes. So NPM1 and WT1 were less common, IDH2 was more common in Black patients. And we were especially intrigued by the finding that the patients had less frequent mutations in NPM1 because of its known association with favorable outcome in AML. So we first thought perhaps Black patients do worse because they have fewer mutations in NPM1. And surprisingly, even though the patient numbers are admittedly small, the overall survival of Black NPM1 mutated patients was notably shorter than that of white patients. And even in the favorable risk group here on the right side of patients without co-occurring FLT3-ITD, which really led us to suspect that the lower frequency of NPM1 mutations in Black patients was likely not the major contributor to the survival disparities that we were seeing. So lastly, as final part of the study, we conducted the union multivariable outcome analysis to identify clinical and molecular features that impact outcome just within the cohort of the Black patients. And in this analysis, we saw that the presence of FLT3-ITD and IDH2 mutations as the only two gene mutations that associated with a higher risk of death in Black patients. And that was important for two different reasons. First, as you all know, those FLT3 and IDH2 have now like targeted inhibitors that could be perhaps used for these patients. But secondly, it's also worth noting that other gene mutations that have a known prognostic significance, such as also RUNX1, P53, they did not remain in this final multivariable model. So in summary, it really became clear that there are differences, or have to be differences, in the underlying disease biology, and that established prognostic features for the AML population at large do not necessarily carry the same weight also in Black patients. So one question actually first brought up by one of the reviewers of our paper, that at that time we did not have the patient cohort size to adequately address it, was whether the survival disparity would also hold up in the very young patients, given their known better outcomes compared to older adults, like saying those that are 40 to 60 years, because they have likely fewer comorbidities and so on. So we felt that this was a pretty good and important question to answer, especially since the AYAs are really a neglected, understudied group, and also molecularly not as well defined. So we went back to the full Alliance registry, and this time we reviewed each chart from 1983 to 2016, which was a total of 816, 102 Black and 217 white patients. And after the somewhat surprising notion, I'd say, that relatively few patients, especially Black patients, had to be excluded due to insufficient treatment, we were able to compare the outcomes of 89 Black and 566 white patients in our studies. Of those, 4 Black and 85 white patients received an off-protocol allogeneic transplant in first complete remission, and we analyzed those separately. And this left then 85, and that left then 85 Black patients, 481 white patients for the survival analysis. And they all got full intensive treatment protocol, and per the Alliance, full treatment means they got intensive induction, and then either they got an autologous transplant in first complete remissions, or 3 full cycles of intensive consolidation. And in addition, 50 Black and 277 white patients had tissue available and were analyzed for their molecular features. And while we saw again no differences in the clinical characteristics between Black and white AML patients, we did find striking differences in the molecular features, starting with the cytogenetics. As you can see here in the pie charts, only 19% of Black AYA patients had a cytogenetically normal karyotype, which usually would be the patient majority, as you know. And instead, almost 40% of patients had core binding factor rearrangements, making it really the largest group. And this alone, to me, is really striking, knowing that all those AML, that the CBFs are AML defining events. And this can and probably should change our current understanding about the epidemiology of CBF AML. And we actually at the moment doing some long-range sequencing to better characterize the structural composition of these cases as well. And also with respect to the frequencies of known AML-associated gene mutations, we saw that there are race-associated differences, including, for example, more KRAS and ASXL1 mutations in the young Black AYA patients. For the survival analysis, we decided to be as granular as possible, and thus we further split the AYA population into the youngest group from 18 to 29 and the slightly older group aged 30 to 39. And strikingly, we found that nearly all of the discrepancy between white and Black outcomes were seen in the youngest group. So the Black patients aged 18 to 29 had a 60% early death rate compared to 3% on whites of the same age, and they had a CR rate of only 66% compared to 83. So the early death rate here is defined by deaths within 30 days of diagnosis, which I think is an important, albeit a pretty unusual endpoint to assess, I have to admit, but it might tell us indirectly something about the very initial care a patient received. And in addition, it also might tell us something about immediate toxicities, for example. So touching on both structural racism aspects and, for example, perhaps drug metabolism. On the other hand, the differences in the complete remission rates were also super meaningful. And I think really need to make us think more about the treatment intensity and modality that we're currently providing. These poor early survival and response disparities are continued when analyzing the overall survival by age and race. And as you can see here, the racial gap in survival enlarges to almost a decade shorter median survival of Black compared to white patients, 1.3 versus 10.2 years. Next, albeit the patient groups are getting really small and small, we compared the survival of core binding factor and non-core binding factor AML patients. And while Black core binding factor patients indeed had better survival than Black non-core binding factor patients, the survival of Black patients with CBF only resembles that of white non-CBF patients. Even most importantly, the survival of these non-CBF Black patients is just incredibly poor, with a 5-year overall survival of only 12%. So that we can basically say that the high proportion of the core binding factors that we see among Black AYA AML patients likely even positively skews the survival of the young Black patients that we have been reporting here. Unfortunately, I know I'm running out of time here, but I wanted to at least touch upon some of the additional molecular aspects. Because as I mentioned before, the mutation frequencies we have been comparing in the studies are those mutations about whose existence we know based on the large genomic studies, which were conducted on white patients. So the focus of my lab is to complete an exome and transcriptome sequencing study of more than 100 Black patients, which we hope to finish before the end of 2021. But for this study here, we at least started with a very small pilot of integrated genomic profiling on paired diagnosis and relapse samples. Here, just examples of 4 patients. And as you can see, known AML-associated mutations seem present also at diagnosis in Black patients, such as NRAS, you can see NPM1, DNMT3A. But we also saw that almost 25% of the variants that we found have never been described in any of the big hallmark studies. The second thing these results suggest to us were that the original clone in the cases we studied seemed almost always present at relapse. And maybe again, at least a small indicator of insufficient treatment intensities. And this is further supported by the fact that we were indeed able to detect NPM1 minimal residual disease at time of morphologic GR in those mutated cases that you see here. So there is obviously lots of further work and follow-up here. With that, I would like to make a very hard cut and just for 1 minute touch upon socioeconomic features. And I would like to do that because I really strongly believe that we need to start integrating genetic and social factors into the same analysis when we are looking at outcome impacts. So what we have been working on in addition with the help of the College of Public Health to make up for missing socio-demographic data on the cooperative group patients, which I think is a problem for all cooperative groups that we are using. And so we used a zip code-based socioeconomic composition calculator, which provides basically a surrogate for the overall economic status of an area. And here you can see when it's darker green, and these are the wherever lines patients are being treated, being area of lowest deprivation. And based on that, we defined two social disparity levels, low and high, and tested for associations with clinical and molecular features. And not surprisingly, patients with high social disparity index were less often white and less often get an allo transplant in first complete remission. And also, being part of this high social deprivation group associated with shorter disease-free and overall survival. And this stayed in the final outcome model in addition to ELM. So this means that residents in areas with this high socioeconomic deprivation associated with poor survival. And I can't show the data here, but what was also interesting was that there didn't seem to be associations between social deprivation and survival among Black patients, which in discussions with the public health people makes us wonder whether systemic racism really outweighs the poverty-associated survival impact. But of course, it also bears a chance because social area deprivation is something that we can address and potentially overcome. So as a conclusion, what can we do? Obviously, there are too many things to list in here, but at least I want to perhaps also help spark a little bit of the discussion and all the points that Patricia already mentioned in her introduction. Diversity and inclusivity is always an important thing to say, and I think it just touches upon so many different things. We all talk about more diversity in clinical trials, but also diversity in treatment teams, meaning there are a lot of studies that show if patients are being treated with physicians, nurse practitioners, nurses, research coordinators of their own ethnicity, that the outcomes are better. We need to be more diverse in sequencing efforts. Now, I want to be very clear that I think we need to get away from these percentage inclusions that we all include into our R1s or whatever grants, where we have to reflect the general population at large. Because AML is so heterogeneous, cancer is so heterogeneous, we will only understand the molecular complexity if we have enough power. And this means we learn so much about the AML from Caucasian patients because we sequence thousands of patients. We need to do the same sequencing efforts, and we can't only do a 5% population-based inclusion in here. The fourth part is that we really have to stop looking at social and genomic factors independently. I know we can't always do it, but at least trying to assess those is an important step. Access is important, treatment trial finders, community engagement, but also early education. And this is something I'm really passionate about, thinking if we want to have physicians, nurses, as what we just said in diversity and inclusivity of minority groups, we need to get them to the best possible high schools. We need to get them to our colleges. And in fact, the genomic studies that I showed to you were done by two incredibly talented high school students who learned over the summer how to do like library preparations and sequencing, who stayed on in my lab and who had really the idea to study the background of Black AML patients and help their own community in this way. And they were both accepted to Ohio State afterwards. And also we need prospective studies, not only to get the socioeconomic and molecular data, but also to really be able to conduct pharmacogenomic studies that I think are really needed to understand better what the associations are, because nothing else what I showed you today, it was nothing else but associations. And also it gives us the opportunity to provide structured support for our patients. And then even more important in the setting of all our targeted agents that we have is that the groups are getting very, very small. And I think it's going to be very important, especially for minority groups, to understand how our targeted agents work. And so what we have been doing over the past year and a half is that we try to assemble sufficiently large data set with a little consortium of other cancer centers where we are uniformly collecting treatment data, molecular data, outcome data, also social demographic data over one central REDCap database, where every participating institution can have like access to these data. If wanted, we can provide statistical support via the center, but just to be able to get to the patient sizes that we need to make like meaningful conclusions on each and on each group. And again, thanking also people who contributed, thanking all of you for listening to me. Sorry if I went a little bit over this time, and I'm happy to take any questions.
Thank you very much. That's great. So I'm kind of like, doing the questions. So hi, everyone. If you have a question, can you, I think we have time for like 2 or 3 questions. So if you raise your hand, use the raise hand function, that would be great. Or, unmute yourself and just ask away.
Can I ask the first one? It's kind of obvious. I think other people.
Sure. Go ahead, Patricia.
So it's kind of what are your perspectives on the nitty-gritty molecular mechanism underlying the different mutations? Do you think those arise because of the person's exposures, or do they arise because of the genetic background, or both?
No, it's totally obvious, and I wish I'd know the answer. I think we have to look at both in parallel. No, I think the easiest, that's why I said I'm super intrigued to look at the TA21s or inversion 16s, because their breakpoint seems like more tangible to me. When I said we have to look in cis and trans, is there anything that makes more prone the area? So I do think we have to look at this, and we have to do this in other factors. But the second part, I don't know. I really think it must be other constitutional genetic events. That's my personal opinion. But I think we need to also look at environment, even though it's way harder to study.
Thanks.
Craig?
Yeah, so thank you so much, AK. That was a terrific presentation. And I guess just to add a little bit to Patricia's introduction, I wanted to point out that AK was featured as a plenary talk for the American Society of Hematology, which is the premier meeting of the year for this field. And you can see why the nature of this work is so important. So just to get in the weeds just a little bit, AK, the striking difference that you showed for young patients was particularly disturbing. And as you know, allogeneic transplant is still, bone marrow transplant is still the best therapy that we have, particularly for younger patients. So is part of that disparity simply getting to this very expensive and medically intense procedure?
It may be. So I couldn't show the data. We were able to look at a few patients, but again, we don't have the power yet. So we only had 4 Black patients that got a transplant. What we can say is that there was no statistical difference in patients of the white patients who got the transplant versus those who did not. But I do think that it will be one of the contributors that go in here. So I think this goes, however, then again, to me at least, along with treatment intensity and consolidation intensity, right? So I think that it goes along these lines of that we at the moment don't seem to be doing a good job in eradicating the leukemia clone, and perhaps the allo does. So I'd be hopeful with larger numbers that will come out, and maybe a good way to change it.
And in interpreting your data, is it correct to think, because of the access to trials issue that you pointed out, that the vast majority of these patients would have received sort of conventional induction chemotherapy, and they're not receiving all the sort of new things that have come along in the past few years?
Yes, probably, yes. And I mean, that will be interesting if we follow up, because the studies from the Alliance, what is nice about them is that they're all uniform, right? But we wouldn't call it trials anymore, because what we basically look at is 7+3. So we don't know anything else in here. And I'm very concerned that we know so little about patients treated, about 7+3 in minority groups that I don't even want to know how we, if we don't combine our efforts, how we want to find out more about patients treated on nowadays clinical trials where we have a way bigger variety.
Thank you.
Yeah, thank you. We'll move on to the next speaker. Patricia, you want to?
Sure, yeah. The next speaker, in fact, can you unshare your slides so Chris can put his slides up?
I'm trying.
The next speaker is Dr. Chris Chenu, who's a professor of bioinformatics and personalized medicine, as well as the director of research for the Colorado Center for Personalized Medicine, which is also abbreviated CCPM. And where's his talk title? Let's see. Implementing the future of personalized medicine and population health monitoring, specifically focusing on the CCPM biobank.
Cool, thanks so much. Just, can you all see my slides okay? Can you hear me?
Yes and yes.
Excellent. Okay. Well, thanks so much for giving me the opportunity to speak today. I think you'll actually see some tie-ins between some of the sort of health monitoring stuff that we're thinking about in CCPM and what Dr. Icefeld was talking about in the previous presentation. Just to give a brief background for those who don't know, the Center for Personalized Medicine now is just over 5 years old at the University of Colorado. It began sort of in Congress with the Division of Bioinformatics and Personalized Medicine. And over the past 5 years, we've deployed a large number of things, including developing our in-house biobank, developing all the clinical infrastructure to ensure that we're properly able to return results to patients, and provide a research engine that allows us to do sort of genomic discovery and personalized medicine at scale and think about new translational opportunities. So as we can see here, this has sort of been until 2020. And so now we're sort of in the space where we're trying to actually like sort of become kind of standardly integrated into care and provide unique research opportunities that wouldn't be present otherwise. So one of the things that's incredibly exciting is that we've been able to partner up with Regeneron Pharmaceuticals, and they're going to be generating data on hundreds of thousands of individuals over the next couple of years. So particularly as people are interested in identifying cohorts, for example, carrying specific BRCA mutations or Lynch syndrome, that sort of thing, there's sort of a pre-built opportunity here to work with CCPM and partner up on some of these initiatives. One of the things that we've tried to ensure from the start is that there's opportunities both in clinical and in research. So keep in mind throughout this, I'm going to sort of be focusing on the research side. That's kind of my interest primarily, but certainly everything sort of has these goals in parallel. So far we've actually been quite successful in recruitment. We're almost at 200,000 participants from the UC Health system. As we can see, they're broadly distributed across the state. Today, I'm going to be presenting on genomic data on about 34,000 of these individuals that we've generated data on so far. But really, it's worth actually thinking about CCPM as a national biobank. Of course, we're centered in Colorado and most of our patients are here, but we have individuals with home addresses across the country. And importantly, in contrast to a lot of other biobanks that are centered only in major urban centers, we have a large number of participants that are living in rural areas where access to healthcare is a little bit more challenging. And that's something that we've been sort of highlighting in a lot of our initiatives so far. So like I was saying before, it's worth kind of thinking about sort of the ongoing opportunities and thinking about CCPM itself as a living resource. The patients that consent to be part of the biobank, they agree to recontact. They agree that their data may be shared either with other researchers on campus or in partnership with appropriate industry collaborators like Regeneron. They agree to be able to receive results. And so everything that we do kind of hopefully can feed back into the medical system ultimately. That's certainly our goal. And just to remind everyone, this data is totally available to anyone who's interested. As you can see here on the right, when you go into Health Data Compass and request different data sets, we are checkbox alongside other aspects of data. I'm happy to sort of walk people through the process and think about the things that can be sort of successfully accomplished using this kind of data. It's totally free to researchers on campus. You know, we have an additional kind of approval process to reflect the best interests of our participants. But it's totally not onerous. And the idea is to get people out there and using the data. And just wanted to highlight on the lower right, Nikita Pozdiev, who's in the endocrine faculty here and also part of BIPM, leads our translational informatics service that you can think of as a way of partnering up kind of like with CETA and other resources on campus to allow people to do genomic analyses without necessarily having all that capacity in-house. And just to describe how we're, you know, part of the sort of ongoing research dialogue, you know, one of the major things that we were interested in participating in was the large-scale COVID work that was published this summer in Nature. And we were only one of 4 biobanks in the US that contributed to this venture. So we're pretty proud of being able to contribute to that. So I'm going to talk to you a little bit about sort of population health monitoring and what we think about that within CCPM, and then sort of close with some implications for polygenic scoring and genetic risk. And just to provide some examples of what we can do with genetic data, one of the major things that we can do, and in fact we have to in statistical genetics, is understand the genetic ancestry of our participants. And like most other major medical centers, our population, our you know, sort of catchment includes multiple populations. And as we can see here, they include ancestry from all over the world. And so as we, you know, sort of think about interpreting that, of course, these sort of blocks are very large. And we want to actually get into understanding what the actual populations are that make up our participants in the biobank. And so we've been looking at a series of methods that allow us to detect relatedness across individuals, with the idea if you can find a cluster of individuals that may not necessarily be immediately related as, say, pedigrees, but kind of extend out to, you know, 3rd or 4th cousins or that sort of thing, that share ancestry over the past few hundred years, these individuals reflect populations that are more likely to share ancestry with each other and are also more likely to sort of potentially share different exposures and be geographically located in kind of similar areas. And so we published a couple of papers sort of describing this approach over the summer. So this was a methodological paper on rapidly identifying relatives in biobank scale data sets. We can take this information then and we can cluster these pairs into larger clusters of individuals that share more affinity within than between groups. And sort of applying this in a health system context is another paper that we published this summer. Kind of thinking about what this means in our data sets. Again, if you're looking at populations from the past few hundred years, we're not talking about these large scale OMB kind of classifications, right? These could be sort of fine scale individuals from a restricted region. You know, if we think about sort of the overall broad patterns of ancestry across Latin America, certainly that doesn't constitute a single monolithic population. And so in fact, when we apply this in CCPM data, we identify over 100 clusters, and these are individuals that have sort of enough CCPM individuals that we can, you know, run statistical analyses on these individuals. And what we can see based on the sort of colors in these different bars is that there's different genetic ancestry in these different groups. But really the importance of looking at this sort of relatedness-based way of thinking about populations is we can identify populations that have similar broad patterns of genetic ancestry, as we can see here, but they sort of cluster differently, and it reflects this more recent kind of history in these different groups. So just as an example, here's 3 different clusters of individuals that are largely assigned in the EHR as Hispanic. They have similar genetic ancestry proportions, these sort of colored bars on the top plot. but they reflect sort of different patterns of recent history. And of course, part of this recent history reflects in current geography. So here's sort of mapping things to 3-digit zip codes. We can identify that certain clusters are over or underrepresented in certain regions of the state. And of course, this starts to, impact potential exposures and that sort of thing, has associations with socioeconomic status and the like. So, one of the things then that we can do, because we have data across the entire electronic health record, is identify individuals that are, or identify traits that are over or underrepresented in each group. And we're going to do this kind of controlling for all sort of standard covariates in our field, with the idea then that this gives us a way to sort of hone in on the specific disparities that affect different populations in our health system and how these different groups are interacting with the health system, in fact. And so, this isn't just necessarily looking at different OMB categories, we can look within different groups. So one of the things that we've been really interested in doing is looking within groups that would be assigned the same race ethnicity terms in the EHR and actually showing that they're interacting with the health system differently. So here's an example of 2 different non-Hispanic white clusters. And what we can see is probably not a surprise to folks in the Cancer Center that individuals in Colorado are at risk of skin cancer. But one of the important things that we see is if we think about sort of quantifying things like tobacco use disorder as a risk factor, it's only found in one of these groups, of course, and we see sort of skin cancer in both, which totally is sort of expected. But it's this idea that, these risk factors aren't necessarily monolithic, and particularly on the research front, we shouldn't necessarily think of them as being as such. So in contrast, here's 2 of our Hispanic clusters. Again, we have a large number of clusters. I pulled out a couple of these just as examples. Again, we see that tobacco use disorder is sort of preferentially enriched in one group, and in another group, sort of aspects of liver cirrhosis and other, potential risk factors that could lead to things like liver cancer are found in another group. And so why exactly all of this stuff is happening, we're still trying to sort of figure this out and be able to overlay the kinds of environmental and socioeconomic variables that we can derive in collaboration with the participants of our biobank. So in the interest of time, I'm not going to talk too much about polygenic scores. I just wanted to mention the fact that we're actually working on a large number of different scores in collaboration with a large number of other studies across the US and the world, and really kind of think about this question of how we can sort of surpass these issues of the sort of Eurocentric bias that's existed in human genetics so far, and how that can impact our patients. One of the things that we've found, and we were the first group to publish on, was this idea that you just get these sort of unpredictable shifts in polygenic scores across different ancestries. And actually being able to identify those differences and sort of standardize those distributions allows you to compare risks on a more sort of apples to apples comparison. Here's just an example in atrial fibrillation, but we can do this for any particular trait. And in fact, this is some work led by my postdoc, Lemon, who's done some really exciting work kind of characterizing the polygenic score catalog and providing it as a resource for people interested in applying these kind of risk factors across ethnicities and looking at their impact. And so just sort of pulling down cancer and neoplasm scores and their impact on different things. Here we're sort of taking a fairly extreme, like the top 10% of the distribution as a risk. And what we find is that there's a huge amount of cancer-related outcomes that have strong polygenic score implications. Especially down here, sort of looking at prostate cancer, that's become sort of an active area of interest for us and other groups across the country. So like I was saying, we're starting to work on this in the context of prostate cancer. Some folks in the Catchment Connections meeting heard from my colleagues at the Fred Hutch about our work on colorectal cancer. one of the things that's sort of a sneak preview for now is we're working on some RFAs to sort of enable other people on our campus who are interested in performing this kind of work in collaboration between CCPM and the Cancer Center. So that should be happening this fall and winter. So just to conclude, we're able to perform this kind of population health monitoring. There's sort of broad implications for cancer, but it sort of gets to this question of what are the populations that you're monitoring in this sort of population health kind of context. And we think genetics, and particularly genetic ancestry, provides a powerful tool to interpret this kind of data. Of course, this has to be done with major caveats, but there's some real potential for some of these to turn into translational insights. And so with that, I'd just like to thank the members of my lab and my funding from NHLBI and NHGRI and the other folks in CCPM, and happy to take a few questions.
Thank you. And I think we have time for 1 or 2 questions. So raise your hand or just unmute yourself and go ahead. Craig.
Chris, thank you. It's been so fun to see the CCPM develop. Things are moving fast. It was particularly exciting to see the portal that you showed where investigators can go in and start querying the database. Just from your experience with recent history, is that at a point where anybody, say me, who knows nothing about GWAS studies, can go in and actually start to ask intelligent questions, or we still kind of need a little handholding, maybe the first time through to kind of do that? And you mentioned that Nikita and others are available to kind of help folks navigate that portal. Is that a fair statement?
Yeah. So yeah, certainly, we could put out a bunch of tools, and if people don't understand how to interact with them, then they're kind of, then they're sort of unfairly useless, right? And so we definitely recognize that. So there's 2 levels. One thing, and I should make sure that this gets circulated to the entire Cancer Center base. So yeah, we have a sort of self-service kind of interactive portal, so people can look up a GWAS of their favorite trait, or you can look up a variant. Let's say you're really interested in some HLA variant. You can ask, like, what of any possible trait in the EHR is associated with that. Okay. So those are available for exploration and stuff as self-service. I'll circulate around the sort of the access for that. That on its own can kind of help generate hypotheses and provide some pilot data and feasibility. But that sort of next level of like, okay, I actually want to do something, and I want to look at, the people with lung cancer who don't have a history of tobacco smoke, or something that's a little bit more nuanced, right? And if you don't necessarily have the capability in your own lab or that kind of thing to run those kinds of analyses, it's totally fine. We have a process in place for people to work with Nikita's team to help make that happen. So it's worth just thinking about. Those are resources that are available on campus. While it's early days, those are free. So we're just trying to ensure that the resources get used by everyone.
So Chris, I think what most people don't have a good sense of is what data elements are typically associated with the GWAS data. So a history of smoking and blood pressure and et cetera, et cetera. So is that actually on your website anywhere so people can help craft those questions?
So we provide a link out to, we use largely for this kind of self-service tool, we use ICD code-based definitions, and we have sort of there's a standardized mapping that people use for that, and we provide links to kind of descriptions of those. Beyond that, particularly when it comes to quantitative traits or things that may be, sort of found patchily in the EHR, that sort of requires a little bit more kind of self-service digging. But all of that's available in collaboration with the Translational Informatics Service and CCPM.
Great, thank you.
Let's take a quick question from Brian. and then we need to move on after that.
Yeah, no, thank you. And I just plug to Craig, Nikita, I've worked with Nikita a little bit on this, and I am not facile on this at all. And it actually is a very user-friendly system. So even a guy like me can use this. That's great. Chris, my question, maybe it's not, it just blew me away a little bit, I think, is when you did your cluster analysis from CCPM and you found 127 distinct groups.
Yep.
Can you scale that up? When we're thinking about ancestry, how many in the world do you think are we going to have of cluster groups? And how are we going to really begin to study them? I mean, that's a challenge.
It's a huge challenge. And yeah, I mean, some of them, if we have like 20 or 25 participants from CCPM, it's hard to say a lot, and particularly how those individuals are interacting with the health system. The number of clusters is probably, exceedingly high, and some of them probably can be collapsed together depending on, the way that we've sort of been discussing it is like particularly from a biomedical perspective, they're as relevant as the data describes potential patterns of disparities and that sort of thing. So one of the things that we've been interested in now, again, because we do a lot of collaboration with other biobanks, is like ways of sort of securely identifying whether the same clusters are present in different health systems or not. So we're working on an initiative with UCLA and Mount Sinai on that right now. But then the goal is, if we can do that in this kind of federated, decentralized model, then there's no reason it has to stop at 3, right? We can look at a dozen health systems or 25 or whatever.
Thank you. Patricia?
Okay, Adam, if you want to start sharing your slides, I'll introduce you. So Adam Green is in my division, yay. Pediatrics, heme/onc bone marrow transplant, and he is an associate professor of pediatrics and attending physician in pediatric neuro-oncology and part of the Morgan Adams Pediatric Brain Tumor Research Program. And I'll let him do his title because it's right up here. So thanks, Adam.
Great, thanks so much, Patricia. You seeing my slides okay? Perfect, okay. Yeah, so thank you so much for the chance to present this morning. So I'm going to talk a little bit about our work on socioeconomic disparities and demographic disparities in childhood brain and spinal tumors in the US. And so we kind of focus the work down to Colorado as we try to kind of move it more to a patient and interventional level. And I'm an MCO member, so I run a translational research lab that works on pediatric high-grade gliomas and how we target these tumors as well. Pediatric brain tumors are now the most common cause of childhood cancer death, even though leukemias are more common, because we're still not doing very well with a lot of the subtypes of childhood brain tumors. And yeah, so a lot of what I do is try to find better targets and better treatments for some of the most aggressive brain tumors that we face. But I think a lot of the room that we have to improve is also addressing demographic and socioeconomic disparities that may also be contributing to the poor outcomes from these diseases. And there's a fair amount of literature in adult cancer, especially in some in adult brain tumors. So there was been a couple studies, including this one, that talked about some socioeconomic disparities like insurance-based disparities in glioblastoma survival. And there's been some investigation into childhood cancer disparities. A lot of that's been focused on leukemia. And so when we started out this work, our goal was really to start to identify groups that were at risk in terms of childhood brain and spinal tumors. So we hypothesized that there were both pre and post-diagnostic factors that lead to poor survival outcomes for pediatric CNS tumor patients who have demographic, so kind of racial and ethnic, and socioeconomic disadvantages. So in this initial study, we used the CR18 registries, and this work was led by Rob Feinberg on left here, who is a CU Med student at the time, and Shadi Zahidi, who is a research assistant in our program. So SEER is a national cancer registry. And we used, we don't really get a lot of patient level socioeconomic information from SEER, except for insurance, which has been collected since 2007. But otherwise what we do is we use census-based information, so we can kind of tell from where the patients live, what the socioeconomic conditions are in their county, basically. And then we can correlate those characteristics with, we looked at metastatic disease at diagnosis and then also at overall survival. And so these were kind of the initial findings. So when we looked in a multivariable analysis at survival as a whole, we found that Black and Hispanic patients had higher risk for death across these models, even when controlled for other characteristics than their white and non-Hispanic counterparts. But when we controlled for socioeconomic characteristics, we did see some mediation of those effects based on socioeconomic characteristics. And so then we wanted to kind of look at, so again, these kind of pre-diagnosis and post-diagnosis factors. And so we wanted to look at, do these populations also have a greater risk for having metastatic disease at diagnosis? And we found here that for Black patients, there wasn't really a higher risk of metastatic disease at diagnosis, but for Hispanic patients, at least in one of our models, there was a higher risk. And then to look at kind of post-diagnosis factors, we stratified our analysis for patients who had localized disease at diagnosis and had metastatic disease at diagnosis separately. And so here we found that for patients with localized disease at diagnosis, both Black and Hispanic patients had poorer outcomes, again, even controlling for the extent of disease at diagnosis. And that was true for metastatic patients as well. So we concluded from this initial study that CNS tumor survival is worse for Black and Hispanic children. And the differences appeared to be come mostly from post-diagnosis factors. So potentially things like, poor quality of treatment, poor access to clinical trials and advanced treatments and things like that. Although there were some pre-diagnostic differences seen, especially for Hispanic patients. And that some of those disparities were mediated when we controlled for socioeconomic factors. So those socioeconomic factors may be playing an independent role as well. Okay, so then another set of work that we've undertaken addresses patients with early death. So Dr. Icefeld mentioned this before, but we define early death as death within 30 days of cancer diagnosis. So, we know that this is a group of children in pediatric cancer who, are really unable to benefit from the treatment advances we've made in the field because they're dying too soon to kind of really benefit from treatments. So we did another STEER analysis of 555 early death patients, and we compared them to basically all the other children, either who were long-term survivors or at least died after that 1 month period. And so these were our results, and we published these in JCO a few years ago. And we looked separately at different disease groups. And so specifically looking at CNS tumors, we found that infants in very young children were especially at risk for early death, as were older teenagers. And it seemed like that was for somewhat different reasons. So the socioeconomic risk factors were present for the older teenagers, where they were not really present for the young infants. And that seemed to be maybe then a difference in the cause of those disparities. And then, we also found that Hispanic children were at higher risk for early death as well in CNS tumors. And when we looked at specific diseases that put kids at risk in general for early death, we found that there were a lot of brain tumors in this group that were higher as early death. We found AML here too, just kind of referencing again back to Dr. Icefeld's talk, whereas only one of the brain tumor study, low-grade gliomas, were in the group that actually lowered risk for early death. So we then, really want to kind of focus. We can identify disparities, but we now want to focus on what causes the disparities and how we intervene on those causes. And we talked some in this forum so far, that there are really complex potential causes for disparities, and it's hard to kind of piece out those causes just using a population study. So we really want to get down to the patient level, and this is just kind of chart showing the kind of complexity of those potential causes of disparities. So we've now we've got a grant through the Community Outreach and Engagement Office through the Cancer Center this past year to kind of take this to a patient level here at Children's Colorado. So we did a kind of a deep dive on the charts of 858 brain tumor patients from our center. And we looked at kind of different sources of data, including we have a patient database. We use our electronic medical record. And then Ali Hilliard, who's pictured here as a master's student who did a really deep manual review of these charts too. So kind of collected all of these data points, including some of those socioeconomic data points that we're unable to get from the databases. And that included one really helpful source of information was the social work notes that kind of identified socioeconomic risk factors for each family when they were first diagnosed. The interesting thing that we found is we really didn't see the kinds of disparities that we were seeing in the National Cancer Database looking at our own cohort. So we did not find that Hispanic or Black patients were at higher risk of poor outcomes. We did find that Asian patients had a higher risk of death, which was interesting. But when we looked at urban and rural, we didn't see a higher risk of either way. And then we looked at some of those social work concerns. And there was, some on univariate analysis, some higher risk for patients with financial concerns, but that disappeared when we did kind of a multivariable analysis. So why is there a difference when we look at our Colorado cohort compared to SEER? So, obviously there are advantages when we do a population study in terms of power, and especially with, somewhat rare disease like childhood CNS tumors, that becomes a disadvantage in looking at kind of more patient level. But it'll also be that some of these disparities, may be ameliorated by, I think what is a strong health system for underserved patients that we have here in Denver with Children's and Denver Health present. So we've also undertaken a patient-level early death study now, and this is Katie Lynn, who's one of our first-year fellows, former resident here in our pediatrics program, who has worked on this with me. So she looked at basically all of the early death patients we've had at Children's since around 1990, and that was 45, and we compared that to an equal-size randomly selected control group of non-early death patients. And Katie did a similar chart review. And so here, what we found is, again, we didn't see a lot of disparities between, based on race and ethnicity, language spoken, health insurance, rural urban status in the cases versus the controls for either all malignancies or brain and CNS tumors. We did see some of those similar things that we found in the SEER study, so that first of all, brain tumors were kind of the most common causes of early death in our cohort, and patients with higher grade tumors and disseminated tumors were at much higher risk for early death than patients without those disease characteristics. In terms of treatment, it's very clear that early death seems to be a problem that occurs mostly before treatment, because very few of these patients, in terms of cases, had received, kind of the same type of treatment that our control cohort did. So they're mostly dying before they can get meaningfully treated. And then one interesting thing we did find is that, again, still early, still small numbers, but this analysis looks at kind of the time from first symptoms to diagnosis. And we would expect that our early death patients, since they tend to have aggressive disease, would get diagnosed pretty fast compared to the control group, and that wasn't the case. So we found here that the mean time to diagnosis overall for our early death cases was close to 30 days, whereas for the controls, it was close to 10 days. A lot of variability, didn't end up reaching statistical significance, but I think it seems to be clinically significant. So I've gone over some of the conclusions of this. And again, one of the causes may be that delay in kind of patients recognizing symptoms and getting to first care. When we looked at the subsequent time points, like getting from kind of primary care to subspecialty care and getting diagnosed, those are all very similar between the two groups. So there are some advantages, again, in these patient level studies over registry studies, but we still have a lot more to learn. We've now tried to kind of move toward intervention. And so this was also part of our community outreach and engagement grant this past year. So I created a talk on timely diagnosis of pediatric brain tumors based on a public health outreach program from the UK called HeadSmart. And then we disseminated this information either via live talks, or we also recorded it and put it on the Cancer Center website for primary providers who take care of disadvantaged pediatric populations across the state. And we actually just were on Children's Hospital's podcast that reaches kind of like tens of thousands of patients, families, and pediatricians across the country as well. And so we're hoping to reach some more people through that. But so far, we've given the talk for over 100 providers. And we do a pre-post analysis of their kind of understanding of the symptoms of pediatric brain tumors. And we have a significant kind of improvements in their knowledge from pre-talk to post-talk. And again, hopefully we'll be able to analyze more about how, kind of the impact this makes on diagnosis over time. So just quick discussion and future directions. Again, there's a benefit, but there's also challenges of moving from kind of population studies, which can be done pretty quickly, and get us lots of subjects to the patient intervention level, which is slower, smaller numbers, especially in rare diseases, but is very important for understanding causes and potential interventions. So we're doing some more work on looking at disparities using children's oncology group registries, which have more information than SEER. We're now working on setting up a prospective early death study where we would interview families who have lost their children very early after their diagnosis to better understand patient level socioeconomics and kind of their path to diagnosis and any barriers to care they may have faced. And then we're also interested in integrating our RNA-seq and chip-based transcriptomic analysis that we really have for most of the tumors in our CNS tumor bank with our clinical chart review and information. And so as Dr. Icefeld talked about before, really trying to look at kind of both the biological features of these tumors along with the patient potential sources of disparities and how those interact.
We're past 15 minutes, so
okay.
we really need to wrap up.
Yeah. Yep. And then just the only other thing I was going to mention is Claire Fraley, another one of our fellows, is working with me on a study where we'll do look at patients who are considering their treatment for CNS tumors and do qualitative interviews with them to understand kind of the barriers to care that they're facing in choosing treatments and how we can try to overcome those. And yeah, so a lot of people have really helped with these studies so far and are helping with the ongoing studies, and the Cancer Center and ALSF have been really helpful with funding. So yeah, sorry to go over, and happy to take any questions if there's time or to discuss later in the breakout sessions.
I think we need to move on to the next talk. Oh.
Okay.
Yeah.
Thank you, Adam. So the next speaker will be Dr. Evelyn Borayo, who is a professor in the Colorado School of Public Health, as well as the Associate Director of the University of Colorado Cancer Center and Director of the Community of Office of Community Outreach and sorry, I keep getting the acronym order mixed up, but
You're okay.
all right, so take it away, Evelyn. I will let you.
Okay, and I'm able to share my screen, I
think so.
imagine. Yes, seems like I can.
Okay, good.
Okay, well, thank you for the opportunity to present. I had the honor to do so last year during the retreat. So I'm gonna try to provide the landscape for cancer disparities in Colorado, and really appreciate all the speakers provided excellent examples of the kind of research that you can be doing, but also that is so key to address disparities. My goal is to just kind of show to you here in Colorado what we consider cancer disparities. So here's a definition that the NCI basically provides when the definition for cancer disparities, and is differences in the incidence, prevalence, mortality, and burden of cancer and related adverse health conditions that exist among specific populations groups in the US. And the reason why I'm reading this is because I think it is sometimes confusing what cancer disparities or any type of health disparities means. While there are differences, we need to really start looking more closely. And my goal today is to show to you that if you just look at differences, sometimes it may be deceiving in terms of how differences can actually hide disparities. We know that those differences are related, as we had plenty of examples, ancestry-related biological or genetic differences. Of course, we know the role of lifestyle, such as, obesity, diet, physical activity or inactivity, I would say, the use of tobacco, alcohol, or viruses like HPV, my apologies. But also not being able to detect cancer at an early stage or not receiving timely and aggressive treatment, as well as what Dr. Green referred to, which is access to healthcare. Let me have some water. So this is what I mean by we have to be careful when we talk about differences. This is provided, but I am so sorry. That shows the incidence of mortality. And if you see at the top is all races, and then followed by white, Black, Hispanic, Asian, and American Indian. And as you can see, white could appear, if we just look at the numbers, that they do have higher incidence and sometimes mortality than other ethnic groups, except for Blacks and incidence, where Blacks have a higher incidence of all cancers. If we look at the data from Colorado, you can see that that's somewhat the case as well, where non-Hispanic whites have an overall higher incidence of cancer, of all cancers, compared to, for example, Hispanic, Black, American Indian, and Asian. Same thing with urban, rural, urban, higher incidence. And then poverty status, we do see here that at least the low poverty counties have a higher incidence. And I would argue that this is also based on mere numbers. We have a significantly higher number of white populations, white, individuals in Colorado compared to some of the other ethnic groups, including Hispanic and Black. But as you start looking at what, more deeply at what are other indicators of disparities, mortality is a good example in which now non-Hispanic white and Hispanic start looking very similar, surpassed by Black in terms of higher mortality rates than non-Hispanic white or Hispanic. Same thing with urban, rural. Urban, rural start looking more similar the ones you start looking at mortality indicators. And then for poverty, you can really see the disparity there, where high poverty counties have a higher incidence, mortality rate than the low poverty counties. Again, this is all data from Colorado, and this is 2018 data. Here is the case for late stage disease. This is another indicator of disparities. Again, you see that Blacks then have higher late stage disease, but the other groups are not too behind from white populations or non-Hispanic white. Again, urban and low poverty versus high poverty. What we have done is try to look at disparities beyond just looking at these indicators independently. But when you start actually combining the various factors, for example, race, ethnicity, rurality, and poverty status, is when we have been able to find where some of those disparities lay in the state or where they're at. So we have these two maps. The map in blue is the disparity index. It shows a disparity index with the darker blue showing a higher disparity. And this combines incidence mortality and late stage disease of the four or of the three higher incidence and mortality cancers, which are lung, breast, and colorectal cancers. And so that's what you see there, where counties in the eastern south corner of the state is where most of those disparities are found. We have done other analysis looking at population density, poverty, and ethnicity. The darker green here indicates where there's a higher density of individuals in the state, and not surprisingly, in the Front Range, you see that is the darker green. Now, the lighter indicates either rural or frontier counties. And in the southeastern part of the state, we have a lot of rural and frontier counties. The shaded areas with the lines indicate where at least 50% of the population or higher live under poverty, and the teal dots where at least 25% of the population are Hispanic. So this analysis basically suggests that cancer disparities tend to overlap and seem more pronounced in areas that are underserved, rural, poor, and where Hispanic Coloradans live in the state. Now, we highlight that corner, but you actually see that in other parts of the state, other counties. If you actually start looking at the Metro Denver area, again, if you just look at the Denver metro area in general, you don't see those disparities. When you start doing some analysis by zip code, that we have also been able to identify some neighborhoods where disparities tend to be more pronounced. So based on this data, the Cancer Center recently identified some priorities, and we're in the process of redefining and including or excluding other things in our priorities. But as of now, the populations that we have identified for the Cancer Center and its catchment area, which is the state of Colorado, are rural, Hispanic, and poor populations for the reasons that I just explained. We also have some of the incident cancers across, not only in our state, but other states. I would like to highlight that melanoma, however, is higher, is the 5th incident and has a higher incidence of mortality in Colorado than in other states. So we have identified melanoma, which is not necessarily one of those cancers that other catchments try to target. Then we have also identified some risk factors. Tobacco, we know, is higher among our youth than compared to other youth across the country. We also know that we have low HPV vaccination rates, particularly among young adults and rural populations. In the general population, I would say, if you just look at adolescents, it's very comparable to the rest of the country, but still very low. So it's in the top 40s, close to 50% of adolescents are fully vaccinated. That's still very low. The goal is to achieve 80% vaccination. We also know that there are low breast cancer screening, particularly for women in poverty, women who are Hispanic, and women who live in rural areas. There's also low colorectal cancer screening, particularly among individuals living in poverty, among Hispanics, and among rural. So this is how overlapping, when we overlap some characteristics in the population where we see some of those disparities being more pronounced. Here is one graph that kind of compares Hispanics to other Hispanics across the country. That's another way to look at disparities, because if we just look at the graphs that I showed earlier, yes, they do have lower incidence and mortality compared to the white population. But if you actually start using other groups for comparison, such as Hispanics across the country, you see that Hispanics in Colorado have a higher, significantly higher incidence and mortality from breast, lung, and colorectal cancers. If you compare that to non-Hispanic whites, you see that the opposite is true, with the darker blue indicating Colorado. For non-Hispanic whites, lung and colorectal cancer have a significantly lower incidence and mortality compared to other non-Hispanic whites in the country. So the take-home message is, don't just go by numbers, but start looking at how numbers overlap and how disparities can be very complex. So as you start thinking about doing some disparities research, start thinking about the various layers of complexity. I really like, the challenge to start including socioeconomic status and other variables in the analysis. I think that is very, very important, because it's just not one layer, but it's the added effect of multiple layers. And this is one example. Some risk factors, just looking at where those disparities are in breast cancer. This is a map that we also produced recently to try to understand how much access these populations in these disparity counties have. So this is for breast cancer. And the yellow dots signify where there are treatment facilities or providers. And again, you can see that's another layer of disparities, is access to healthcare, where not surprisingly, the majority of providers, including the cancer center, is in the Denver metro area. But if you look at the southeastern corner of the state, there are almost no providers, and definitely not with the type of treatments that we can offer, for example, at the cancer center, or access to clinical trials. So that's another layer of disparities, is just not looking at, yeah, the disparities are significantly higher in that corner, but access to treatment and other services, because we have also looked at other services such as cancer prevention, early detection, tend to be lower in those remote counties, particularly the poor, isolated, and where a large percentage of ethnic minorities live. Same thing with lung cancer. Again, we were just trying to compare that. Somewhat you see more in the western slope, south area of the state, but the patterns tend to be pretty similar, with that part, particularly that part of Colorado, the southeastern corner, being significantly affected by disparities. I tend to describe our state as if you were to cut it horizontally or vertically, you would see the disparities just with that, with the Front Range kind of being in the middle of that. But a lot of our rural and frontier counties significantly affected by this. And what I want to do here is just to also touch on some of the risk factors, and this is all population-based, lifestyle factors. We know that certain factors put individuals at higher risk for cancer. And we see here that Hispanics tend to report being current smokers higher than the state. The 13.5% on top of current smokers is the state, while compared to non-Hispanic whites, they also have a higher rate of tobacco use. They also tend to be more obese compared to the state, and non-Hispanic whites. They're more likely to report to be physically inactive compared to the state and the non-Hispanic white population. When it comes to early detection, colorectal cancer screenings, they tend to have a lower percentage of screens of colorectal cancer screenings. Same thing if you look at rural urban, they tend to have lower than urban areas. And mammography, as I had mentioned earlier, also tends to be lower among Hispanic and among rural and frontier individuals in our state. And this is my last slide. I just wanted to show you that the challenge that we have at the Cancer Center, not only for basic sciences, but across all the programs, including cancer prevention and control, which is where I'm a member of, where what this map shows you is where the majority of our patients come from. Our majority of our patients come from the Front Range, and the darker blue just kind of indicates where there's a high concentration of those patients that come for treatment at the cancer center, where 80% come from urban areas, about 8.6% from rural areas, and about 10%, not 11% come from out of state. Hispanics are significantly underrepresented in our cancer center, were 7.9%, and after COVID, those numbers were even lower, closer to 5%. That of the patients that we see. The numbers that you see there, the 15, 10, 9, 10, and 11, kind of shows some of the programs that are not treatment that we have in the various regions of the state. And some of this overlaps. So some of the programs that we have in the Front Range may also be present in the Eastern Plains. And these are things such as any type of cancer prevention type of projects, physical activity, smoking cessation. I myself have a couple of projects on HPV vaccination, trying to improve HPV vaccinations. So as a cancer center, not only how we treat patients, but overall, our goal is to start serving our catchment and addressing the disparities in our backyard. Now, the challenges, and I will let Dr. Gino talk about this, is the representation that sometimes we have in our biobanks. And this is not just specific to Colorado. I think this is just in general a challenge of how can we improve that. And I'm going to end up here, but I just want to encourage you to think about cancer disparities research. You don't have to be a cancer disparities researcher to do cancer disparities research. We can all benefit from the type of research that you do. Just start considering looking at disparities. Funding is there from the NIH, so that should be an encouraging, motive that if you do research, you can, get funding. And that ends my presentation. And I see, James, you have a question.
Dude, thanks, Evelyn. Great talk. So I'm really curious about the lung cancer incidence because there seems to be some disconnects with smoking. So Colorado has substantially lower lung cancer incidence, but higher smoking. Hispanics have, relative to non-Hispanic whites, had lower lung cancer incidence, but higher smoking, and even higher other factors like obesity and inactivity. So anyway, what are your thoughts on that?
I think it's an overlapping effect of risk factors. We also have high radon levels in Colorado. So some of the lung cancers are driven not only by smoking, but high radon levels. We have particularly among underserved populations and rural areas where the home mitigation systems are, sometimes non-existent. So I think when you look at cancer, particularly lung cancer, is more complex than just looking at smoking. Is, as you mentioned, high obesity rates in some populations, higher smoking, low access to early detection, cancer prevention.
Those all go in the opposite direction, though, because the higher, we have, if you can add on radon, and that should only explain why Colorado should be higher in lung cancer incidence, not lower than the rest of the United States.
That's true. Overall, we tend to be healthy. If you just look at the state overall, we are healthier. We have more access to healthcare. I mean, Colorado has a lot of things going on. Is when you start looking at subgroups that you start seeing those disparities. But overall, compared to the rest of the country, we are doing better. Now, unless you start really looking at those differences across groups and the multiple layers of poverty, rurality, and ethnicity, is when we really start seeing those disparities.
Yeah. All right. Thanks.
Mm-hmm. Go ahead, Craig. Last question.
Evelyn, something occurred to me that we never talked about, and I wanted just to throw out for discussion, especially for this group. So if you look at Hispanics, in most cancers, they have a higher incidence, and as we had nicely highlighted by AK's talk, there's a complex set of reasons that can explain that. But there's a couple of cancers, as James was saying, like lung cancer, they're lower in Hispanics. And that's in spite of all the disadvantages they have in terms of access and so forth. So do those highlight research opportunities for basic scientists where there's perhaps a biological or intrinsic reason they get less cancer. And have you ever heard, or has anybody ever heard of that actually being useful to better understand some of the underlying pathogenesis?
You know, that is definitely not my area, but I would argue that we just need the research to answer those questions. And the problem that we have is sometimes we don't have that kind of research to say, yes, it's genetically driven, or this is what it is. So I don't want to say that it is driven. We just don't know in many cases. And of course, the rest of you have more expertise in, but that's what I would argue.
Thank you.
Thank you. All right. Go ahead, Patricia.
Okay, so final speaker in this session is Dr. Sean Davis, physician scientist, recent Coloradan, and is one of the associate directors in the Cancer Center, as well as the deputy director for the new Center for Health AI, which he's going to tell us about, I think, today.
You have to unmute yourself if you're going to talk on Zoom, just in case anybody was wondering. Are you all seeing my screen, my slides at this point? Awesome. All right. I know we're a little bit behind, and this talk is going to be a little different than other talks. There's essentially no science in it. That's by design. And this is really to give a fairly high level view of some of the investments and some of the new resources that have been that are coming to fruition on campus. And my hire and a couple of other recent hires are part of that process. So as my title says, this is just an overview for folks who want to go through the slides and read the stuff that I'm going to skip over. The slide link is in the bottom right, and that should be available over time. My contact information is here as well, so feel free to reach out. You know, like every other institution in the country and probably in the world, there's a changing landscape in how we do biomedical research, how we approach biomedical research. It's becoming more and more data-driven. These are things people know. And in order to be successful, we need to build infrastructure, invest in people, and you know, all the kinds of things that MCO has been thinking about for now decades. We're trying to apply in the data science realm. So building training programs, pipelines for pulling people in and doing it in a diverse and equitable way. These are all sort of things that just take a different twist when you're talking about data science. I'll just mention a couple things on here that I wouldn't have been able to say a few years ago. One is that multimodal data analytics are not just a research effort at this point. They're being applied in every large academic medical center and some even clinics to improve care and improve health. And the other is that NIH and other funding agencies are beginning to see the light in terms of investing in research and research in the infrastructure and in the research process, data-intensive research process. Recently, we applied for a so-called Bridge to AI grant. And that grant is really addressing a very particular problem: how we apply artificial intelligence equitably and fairly in the research environment. And we can talk about that offline if folks are interested. This is a sort of guiding principle that world's most valuable resource is no longer oil but data. The guiding principle is not this. It's that data are not the new oil. Data are very different from oil. Oil, we use it, it goes away. It impacts lots of things negatively. Data, on the other hand, are infinitely replicated. We create data. It only increases in value the more we use it. So it's one of those resources that it keeps on giving. Using data typically does improve information, and therefore improves our ability to create knowledge and wisdom. Leaders who are used to traditional resources, and that's a lot of us, think of owning resources, owning data resources, or owning resources in general. And an interesting paradigm, starting actually with NIH and journals incentivizing data sharing through omics data sharing resources, omics repositories, has led to data being treated a little bit differently. And we can also think about it differently. Data have fixed costs to create. So it doesn't really make any difference how whether you're sequencing 10 or 100 or 1,000 samples. The cost to create, to sequence those samples is pretty much fixed. But the value of those data is only increased. The only way we increase the value of those data is to have them used and reused. We can often pay for data once and then share it or distribute it across campus or across the country for that matter. We heard some great examples of the SEER registry being used, and the SEER registry is one of those data resources that's really, it's got a fixed cost to create, and only by folks using it do we actually see the value of it. Curating and integrating data is a really valuable aspect of data resources. Often we want to ask questions, and we think we have the data resources to answer those questions, but those data resources are not easily relatable or integratable, and that leads to the need for infrastructure that supports shared analytics and data integration efforts. Where campuses struggle, I'm not going to go through all of these things, but I'm going to mention that fragmentation of data, people, infrastructure is a big challenge. I will say that the answer to integrate or to fragmentation is not total coordination. There's a reason that things remain fragmented, and there are silos that we have to respect and then work within to get things done. But fragmentation without recognizing its impact on the organization is a problem. Data-intensive infrastructure is also quite different than the investment in it is quite different than the investment in other traditional infrastructure, physical plant, those kinds of things. Wet lab space is more expensive to build and operate, obviously. The equivalent expense of physical plant is compute, storage, data, etc. And one of the biggest challenges for organizations that are moving to a new level of data maturity is to recognize that this infrastructure is equivalent to buildings. So we do need to invest in it in the same way that we think of investing in buildings. We build them for the long term. We support them at an institutional level, and we recognize that it has strategic value to invest in people and this kind of infrastructure that supports the entire campus. Data infrastructure can be shared by large groups of people and potentially even whole regions. For example, the province of British Columbia has a shared compute resource, the entire province. So those kinds of things are things that are valuable, and they're quite sustainable. And finally, the return on investment for information infrastructure can be huge. At NIH, for example, we maintained a very large shared compute system. The investment was about 0.5% of the NIH's internal budget. But it supported between 6 and 10% of the research. So you can think about that return on investment. It's huge, and those kinds of things are things that we've typically, places that are at the lower level of data maturity haven't really begun to realize yet. People is another one. I think everyone here who has data recognizes that finding and keeping good people is challenging, and to build an infrastructure, to build an organization that is going to be successful in a data science endeavor, we need to think about workforce development, training, incentives for people to stick around. And we need to think about that not just in the research space, but also the administrative space, the business space. And we need to extend those pipelines all the way back if we want to be equitable, which is something that the University of Colorado really needs to focus on. We do have a catchment area. We need to extend those pipelines back to fairly early in training and be sure that they're capturing our population in an effective way. I'm going to move along here. One of the things that Casey, Melissa Handel, and I, will get to the names in a second, realized when we first got here, or when we first started talking about coming here, people were explaining to us that there's just not a lot going on. There's a lot of work to do at the University of Colorado, and to our surprise and happy surprise, there's a lot of really substantial and successful initiatives that already exist on campus, and many of them are growing, and they have the resources to grow, the community and institutional backing to grow. So we've had to figure out sort of how those pieces fit together and how we can help them to be more successful. And they've also taught us a lot about how we can be successful on campus, or how we might be successful on campus. The three new initiatives that I've been sort of dancing around, in addition to the initiatives that are on the slide from before, and I left a ... there because there are many others. The Center for Health AI is being headed up by Casey Green. He's from the University of Pennsylvania. Really very dynamic guy. For any of you who have met him, he's delightful to talk to, very creative scientist, and just a really good leader. I'll get to definitions in a bit. The Chief Research Informatics Officer is a relatively new position, generally broadly across the academic medical centers. But it's a role that complements the traditional Chief Informatics Officer and Chief Security Officer. So we'll get to the definition in a bit. And then the third is something that doesn't exist yet, but is in the works, the Department of Biomedical Informatics and Systems Biology, DISPI. Don't take notes on that one because that name will likely change. But the idea is that the Center for Health AI will slowly evolve, or actually will, the faculty who are being hired through the Center for Health AI will end up in a new department, in this new department. The CREO, I'm not going to go through the words on the slide, but you can think of the CREO as the Chief Research Informatics Officer and the office associated with it as maximizing the research value of all the data that live in the institution. That could be business data, it could be hiring, it could be individual data sets being generated by individual labs, or it could be the EHR. And the CREO's job is really to help organize, coordinate, and maximize the value of those data through governance, processes, policies, etc. The Center for Health AI is really, you can think of it as both a community and an institutional organization. It's designed, or the idea is to foster this thriving community of researchers on the Anschutz Medical Campus and beyond, who are inventing and deploying advanced analytical approaches. I would add to that that also includes a community of people who are impacted or who are interested in those approaches and applying them to their own research, but not necessarily deeply involved in developing and deploying such methods. And then lastly, the Department of Biomedical Informatics and Systems Biology, that is going to be the academic home for new hires and potentially some folks who on campus who want to move over and join this group. this group will focus on biomedical informatics broadly, artificial intelligence and machine learning, knowledge engineering. So how do we take information and organize it into knowledge, and analytical and mathematical modeling. So statistical approaches, mathematical approaches. There's lots of opportunities for joint hires. If you're interested, contact me or Casey. There are new training. We're going to need to create new training opportunities. That's already sort of started to happen a little bit. And building a workforce starts with faculty hires and extends from there. This is sort of a bare bones effort right now. We don't have a lot of folks who are directly involved in these efforts, but there's an ongoing recruitment. For those of you who don't know, the link is on the slides. That's an open rank faculty position, and those folks will end up in the department. We have hired one person. He's a junior investigator from the Broad Institute. Here's his website, brand spanking new, 3 days old. But if this looks interesting to you, or if you want to reach out to him, I'm sure he'd be happy to engage with you. He will likely become a Cancer Center member. His research is cancer focused. I'm going to skip over the slide for time, but there's some infrastructure work that it's foundational to Cancer Center and more broadly. For those of you who want to get involved early on, the Center for Health AI does have a membership program now. It's not quite as sort of formalized or as the Cancer Center membership program, and we do have affiliate memberships for people who just want to follow along, want to be involved at some level. And I'd encourage those of you who have trainees to consider going ahead and putting an application in. One of the first things we'll be doing is a lecture series, and we want input from as many people as possible. We're using Slack as our communication mechanism. It's, for those of you who've used Slack, you know, it's, some people like it, some people don't, but on campus, email communications has been really challenging. I think a lot of you would recognize that. So we'll have email lists, but this is a much more facile way of building community. I'll stop there, questions and comments, and I'll be joining, I think, one of the breakout sessions. I'll be running one of the breakout sessions, so we'll have a chance to talk more later. Thanks for your attention.
Great, awesome, thank you. I think we have time for maybe 1 or 2 questions. Or you can also wait until the breakout room.
I have a quick question, actually. It was a great talk. I'm curious about your thoughts when I think about data, in addition to like all the genomics data and everything that you put in, what your thoughts are about the best approaches to have the best possible like longitudinal patient data. If we want to think about like translational research, how to facilitate that patient data, or like especially outcomes, thinking of treatment responses to later on correlate them with being up to date and any of these aspects.
Yeah, it's a great question. One of the amazing things about the University of Colorado, I didn't mention it specifically, but it did show up on the slide, is Health Data Compass. Now, I'm sure that some of you who have tried to work with Health Data Compass, it's not like your traditional data resource. You go and you download stuff, you look through the stuff, and then you download it. It's a complicated beast to work with, both from a social aspect, it's a social challenge to work with it, and it's a technical challenge to work with it. Working with Ian Brooks, who's now leading Compass, one of the focus areas for them is to work with the Cancer Center. And there are a couple of, probably 3 efforts, and I can say it in this small group. So the 3 efforts that we're working on are of integration with a company called Flatiron, who is going to take information out of Compass, out of essentially the electronic health record, abstract it, and give it back to us in so-called well-formed data models. And those data models are fit for purpose for studying particular kinds of cancer. There are 20 or 22 of them. So Flatiron and Compass, that relationship is a work in progress, but that should help some. The second is integration with genomics. So there are some relatively young cancer genomics efforts separate from CCPM that have been ongoing quietly. So this is Kamoko Dara Eisner's work, and also data return from Foundation Medicine. We're trying to integrate those. It's a long process just to figure out really what the processes are for doing that integration. And the third is around changing how Compass works, changing Compass from sort of a freestanding, homogeneous approach to doing clinical research with EHR to a more heterogeneous, fit for purpose set of teams. One of those would be in cancer related, another would be OB-GYN related, for example, another might be pulmonology related, cardiology related. So these groups would develop not just the abilities to deliver data, but also to interface with researchers in a very liaison sort of role to help folks like you and like me who don't do this, haven't done this for a living and want to do it more effectively, and to transfer their institutional knowledge immediately to us. So if the question's been asked before about longitudinal studies, they can interface either with other groups who have done that kind of work, or will have done it themselves with another researcher who's asked a similar question. That's a long-winded answer, but I hope that helps.
Yep.
And I don't know if there are going to be any real specific questions, but I think like the really basic one that that maybe we should start with is, like, I'm a person who works on process or gene X, whatever. How do I start? And I'm not going to become a disparity researcher because of my expertise, but like, where do I go? What's the flow chart for me to figure out how to apply my work to a quest, or if it's already relevant and I just don't know it,
Mm-hmm.
Yeah. So for those of you guys rejoining us, I think we decide to stay all in the same room.
As long as we don't go back up to like, whatever we had before, which is like about 40. So
yeah.
I think we're going to be around 20. So we might as well just stay in the same room, if that's okay. Yeah.
Yep. And Tintin, do you want to start with any questions, follow-up questions for those people who we had to zip through their Q&A time?
Yeah, I actually had a question for Adam, and it was really interesting talk that I think that when you were looking at the Colorado population, some of the factors didn't carry through, and you thought that was because of better care in Colorado. Did I get that right? And
yeah, I think that it's a great question. I think it's one of the things that we think about, right? I mean, because obviously it's a much smaller population, so we have less power to see if there are differences in the outcomes for racial and ethnic minorities and based on socioeconomic factors. But we weren't really seeing even too much of a signal. So again, yeah, I've been part of the kind of health system here over the course of my training and for years as faculty now. And I think, having trained in the Denver Health and children's systems, I think we do have strong care for the underserved population here that and strong primary care that feeds into that system. And so, yeah, and that's just one of the, postulating that might be contributing. But again, it's hard to tell at this point, and we're going to do more analysis of kind of different outcomes as well besides just survival, and going to try to incorporate these biological analyses of the tumors too. So hopefully that will drill down more toward what may be responsible for that difference in our results between the children's analysis versus the national analysis.
Right. Yeah.
I also had a question about that relates to what Chris talked about as well, which is the number of subdivisions and how one reports race. So do you think there's a difference in the national SEER database reporting of race versus what you guys do? And do you guys think that the absolute more genetic definition would help with that?
Yeah, I think it's always something we think about. I think so. Race and SEER tends to be self-reported. So it's basically what the patients tell the hospitals, and then gets reported to the registries. And it's really similar for children's. Yeah, I mean, I think I mean, we always think about race as, it's still a social construct, and I don't think it has, kind of strict biological definitions. So I think that's probably the right the right way to look at it. But yeah, I think it does, make it so that it's a little bit of a challenging variable.
And Chris, it seems like you have some answer too.
Yeah, I mean, there's a huge ongoing question. It's been sort of a major dialogue in human genetics on how to address it and what labels to use, and how does that interface with, like, the recruitment that was done and stuff like that. I mean, one of the major things that like we've decided sort of in collaboration with a couple of other biobanks is to sort of make sure people understand when we're doing this with EHR data, that it's clear that our race ethnicity variables are coming from the EHR, and that's not necessarily self-report. And those values sometimes change over time and that sort of thing. There's actually a National Academy of Medicine task force that's being put together right now, specifically on the use of race and ancestry terms in genomics, to try to address these kinds of concepts. But yeah, I mean, I don't think one would necessarily, the kind of genetic ancestry that we're seeing is not necessarily a substitute for race in terms of thinking about, like, sort of capturing various aspects of structural racism and stuff like that. We wouldn't want to sort of downplay those kinds of effects. So
I guess I'm coming from the point of view of the molecular driven research lab.
Yeah,
they're not able to really address the structural racism issues, but if there is something to be discovered that's really genetic basis, you want to know that you're having reliable data. And I've seen like some lymphoma studies that report ancestry. Again, I don't understand the methods used to say what % African-American ancestry, but they'll set a threshold in their methods of like 90% and say this is binned based on this definition of genetics, and a mutation like our keynote mentioned would happen more frequently in that group.
When you talk about like EHR versus self-reported, what are usually the other sources of that information to an EHR?
Well, we get a sort of derived variable from Compass on this, and so it's kind of a one-shot kind of thing. And we've seen from other health systems that these values tend to fluctuate over time. If people have been involved in a health system for a while, there's opportunities that someone else has checked a box for people in intake or that sort of thing. So, I mean, we just want to be cognizant of the fact that there aren't guarantees that, like, we're necessarily using the exact labels that patient would be using in 2021, I guess.
Yeah.
They're largely concordant, of course, but some of them do fluctuate over time.
Okay.
There's a question and comments from Lisa in the chat. I think one huge driver for the data is actually collecting non-European descent, non-white data. How do we incentivize individuals to participate and allow their information to be used? historically, POC data and samples haven't been treated well, and this has created a huge fissure in research and target population. Are we working to break this down? How so? How are we working with our target audiences, especially to loop them in? This goes towards, like, some of the efforts that the Cancer Center is doing to increase the participation in clinical trials, right? That continues to be really challenging. So I don't know if there are any answers to this. Anybody?
I think
so. This is.
Oh, yeah.
Go ahead,
Sorry,
Sean.
James.
No, Sean, you go.
I was going to say, this is one of those rare situations where we have pediatricians outnumbering adult oncologists. And, for us pediatricians, the obvious answer has nothing to do with targeting underrepresented minorities. It's just increasing adult accruals broadly, right? If we only are capturing, 15% of potential adult participants in trials, whereas in pediatrics we're more like 50 to 60%. We have a very large, gap here, and I think that Colorado is kind of nice in the sense that we do have an integrated EHR across the UC health system, and we do have at least conversations ongoing with Denver Health and other sort of safety net hospitals. So I think there's a lot of opportunity just to increase in more broadly our engagement with patients, and to understand why every patient who is eligible for a trial isn't engaged, isn't being recruited, and that those will extend to underrepresented minorities and other groups as well. But it seems to me that our largest gain would be in just increasing, understanding why people aren't recruited, and then addressing those things for every individual patient. Equitable here is for every patient, not just for folks who fit into specific boxes based on race or ethnicity. So,
yeah, I know for Hispanics, where we particularly underperform, where we recruit less than the number of cancer cases. We have now, we're investing in bilingual clinical coordinators, making a lot more material available in Spanish. We're doing a lot of outreach to different community groups, which I think is huge. Outreach to doctors throughout Colorado who might refer patients to us. And I think, even a lot of just education of people in the clinic, because there's a lot of, for example, if somebody comes in and doesn't speak Spanish, they're not even offered the clinical trial, particularly if there's no material in Spanish and so forth. And so we've got to really do due diligence to make it freely available.
Yeah, I mean, it's the same, on the CCPM end, like our consent form is translated to Spanish, but it still exclusively sits within my health connection. So while that is English speaking only, that's definitely a major barrier. Yeah, I mean, it's, again, it's a major issue in human genetics and these kind of biobanking studies. And, one of the things that we've tried to do is point out how much you can do with the existing data, as biased as it is. You're starting to see kind of more of that in other data sets as well, like UK Biobank and that sort of thing. Some of these large national initiatives where people often kind of subset to the white British component of UK Biobank, and that's it. You're starting to see more people kind of, recognizing what you can do outside of that. So, but yeah, I mean, I think to James' point, the idea, being able to make those connections with advocacy groups and stuff makes a huge difference.
And it's too bad Evelyn's not in this group because obviously she's really leading the way on that. And Chris Liu is doing a ton of work on this.
Yeah.
And so many other people, Sean, others, there's
Mhm.
a lot of people working on this.
Yeah, Evelyn had a conflict, but we were kind of sidebarring about the specific issue throughout the discussion today.
I wanted to, since we got Chris on the phone and Patricia led off with a question, I wanted to come back to something that's come up a few different times in different contexts, and the idea is assuming that we don't have perfect race and ethnicity information now defined at a genetic level, what do we need to do to get that? And this came up, for example, in cell lines, right? For cell lines, we don't typically have that written down. Chris can tell me what the state of the art is, but typically with something between 50 and a few hundred single nucleotide polymorphisms, we can pretty much certainly to the level of what would show up in an EHR as race and ethnicity, we can define specifically any genomic resource. So if we have RNA-seq, for example, we can define that. If we have ChIP-seq, we can typically define that. So I think if you're thinking about, Patricia, how you could address these things at the level of an individual gene, if you have genetic data, we can actually attach to it a fair amount of information that doesn't come as part of the the record.
Let me ask about a really specific example. So I didn't realize this, but like the data that Alexis showed is short sequencing bits, right? So could she take that data and infer ethnicity from patients that she knows nothing about? Just using a TAC-seq or ChIP-seq data?
If she's doing it at a whole genome level, which is I think what she was describing is where they are now, then very likely yes. If she's doing it for a PCR enriched region right around PDL1, then no. But yeah, to the extent that we have anything that's approaching a few hundred variants, and I'm not talking now about somatic variants, but these are very common variants. They're easy to pick up even with one read. You can probably identify with a fairly high certainty where these patients come from. And to get back to James's question, one of the things that we're doing with the Komoko data is to start to expose some of that information. So in that case, we have somatic variation, but we can pull from that, from the raw data, pull from that an additional variable that we can compare to, for example, the information in the EHR. And to come back to that question, what people report, what shows up in the EHR and what is in their genome are three different things, right? They're three different pieces of information and then measure different things. So a self-report is, even if it's quote unquote wrong genetically, is really important. And that's especially in the United States, where that corresponds or that correlates with so many sociodemographic variables that we can't measure. It's really important to get that information. Think of it as something separate. So.
Yeah, because I think in some ways, at least from totally anecdotally, I've known a number of different people who discovered, particularly after they did 23andMe, that, for example, their parents had, for example, they were from South America. The parents like, oh, no, we're Spanish, 100% Spanish, completely European. And they do 23andMe and they're like over 50%, American indigenous. And, and the person told me, I've always looked in the mirror and go, I am not just Spanish, right? But, it's because of that systemic racism where being more European was less likely to be discriminated against. So in other words, you can totally forgive the parents for doing that because there was a good reason for doing that. But it probably leads to a lot of underreporting of non-European ancestries.
Mm-hmm. So kind of given that, like you can go and look in ATCC and look at the cell line information and get some of this. is it worth reassessing some of even the common resources that we use, as well as some of like the patient samples and so on? It's something that Patricia and I talked about, is providing funding to the labs who want to investigate the ethnic origin, ancestry information of their commonly used cell lines that they're using. What do people think about that?
Well, I'll just say if the data exists, that is, if the sequence data exists, we can answer the question. What people do with that information is a different question, right? Sometimes it's useful to know that from a novel, innovative research perspective. A lot of times, actually, unfortunately, it's not, right? There are a lot of, so if you start digging through the literature and asking very detail-oriented questions about what do we know about sort of lung cancer genomic disparities, there's a fair amount of literature there. So some of it is just some of it is getting information about resources that we have. A lot of it, though, I think, is just as researchers, we need to be cognizant or open our eyes to some of the research that might not be in our sort of traditional lanes, and make sure that we're branching out. I put a couple of articles in the notes, the Google Doc. There are hundreds of such articles, but the top one is a little review article about genomics in cancer and health disparities. And there are some efforts at the NIH and HHS levels that are going to help with these kinds of things. But I think a lot of it is, as individual creative researchers, just having a little bit of reading time really associated with what's going on in my field that's disparities related would be a win for all of us.
I mean, some of the obvious things we came across when investigating this is one, that people use a lot of cell line models and did not know, like, let's say 100% of the models you're basing your research on are Caucasian. Well, you know, what discovery efforts you're doing there, you know, could be limited by, of course, you will never discover anything that's enriched in African Americans if you simply do not have any models that represent the US generally. So that's one reason why we compiled these cell line resources for things that are common. But I guess the question became a little what Tintin was asking, is it worth all of us finding out, you know, what are the things that we use, and what do we do with the information which you brought up? So you know, I assume we can go to you guys with advice about, like, is this information too small to do anything with, or is knowledge that my panel of cell lines represents, you know, this array of genetic diversity, is that better than if I just had, you know, all Caucasian prostate cell lines, for example?
I think it's just one way that people can start to incorporate this thinking into their work, right? I mean, because I think, you know, so for instance, we have a bunch of DIPG cell lines now, right? There are probably 30 of them out there that are commonly used. And which ones people end up using depend on a lot of factors, you know, how well they grow, how well they, you know, represent various genetic variations. But if we knew that, yeah, like if we knew the race ethnicity of the patients that those came from, that would be another factor that would be important. And I think it'd be, yeah, I mean, I think it'd be one more thing that we could understand about those, and understand about the diversity of the implications for the data that we're deriving from these models. I mean, yeah, I think it's, Sean, are you like, are there, you know, kind of large analytical papers looking at just the kind of race and ethnicity of all these cell lines for different disease types out there?
So I dropped it in the
oh, okay.
Yeah, and
got it.
So
okay.
yes, there are. And for creating such resources locally, like I said, if we have genetics or genomics on those cell lines, we can probably fairly likely assign you know, an associated so we can take, for example, Chris's population map in a sense and assign those cell lines to those individual groups.
That's great.
Again, not with 100% certainty, and I don't know, we'd have to work out the details, but Chris could tell me whether he thinks that's a good idea, but I think it's probably relatively doable.
And on
Yeah,
we'll add this stuff to our list, but I just mentioned that I've added Chris's core facility as well as some other resources on our curated list. So.
Yeah.
Yeah,
So
I put it in the notes here as well. Yeah, so Patricia's talking about a core facility on campus that can do this kind of genotyping, and we can figure out sort of how to tailor the analysis to the kinds of questions that would be relevant here. Sort of focusing on genetic ancestry, but that should be totally doable.
Can I ask about the next step? So with all this information, is it possible to have a centralized cancer center database where someone can just go in and look up their cell line, or look up, let's say you want to, do an experiment that has 10 African-American cell lines and 10 Caucasian cell lines, and go identify them so that you can go and use them. Is that allowed, if you will? It's not too much private information. And can we set up something like that as a cancer center, resource?
I can try to answer. This is an aspirational answer, but what we did in my last group at NCI was we collected. So we had a very similar setup. We had a couple of core facilities. My group was one core facility. We weren't really a core facility, but we treated ourselves as such. And we had another core facility that did sequencing. And all data that went through there were subjected to some basic quality assurance. All data were sample information about all those data, who submitted them, rough information about what the sample source was. Those kinds of things were tracked in a database. And then the resulting genomic information at multiple levels was also captured and cataloged. I won't say database because we didn't build a database or a set of databases to answer all questions. But when questions like this came up, we had access to the data, which is the first big step. Right now, the way things kind of work at the Cancer Center and actually at CU is that individual investigators submit samples, the data are returned to them, and we don't have any visibility into, we're not building a virtual biobank, essentially.
Mm-hmm.
So my focus right now is for the Cancer Center is on building some foundational capabilities with respect to infrastructure and personnel, so that when we have questions like this that come up, we can answer not that we will build it, but here are the resources that are in place that would help us to build it, and here's what it would cost. We don't have even the ability to walk through that project planning process right now. But from an aspirational perspective, I would like to see all of the data that go through the cores cataloged in a way, not to address your question specifically, Tintin, but if we wanted to address your question, we could say, either we have the data available or we don't. And if we do have the data available, here are the steps that we would probably need to take in order to either build a resource or to address your specific question. And I'm not talking just about genomics. I'm talking about, for example, the animal imaging shared resource is amazing in the sense that Natalie has kept all of the images on disk drives sitting behind her desk for 12 years. So we have the catalog, but we really can't use those data. So that's a sort of low-hanging fruit from my perspective, is to make those kinds of data available too. There's a couple of other details, shared identifiers. The Cancer Center and CU in general don't have a shared identifier. So a mouse is submitted for genomic sequencing or imaging or metabolomics, proteomics, spatial transcriptomics. That mouse gets a different identifier. Even if it's the same mouse submitted to 5 different shared resources, we'll never be able to tie those data together without a shared identifier. So the concept of having a common identifier across the cancer center is another sort of aspirational but foundational capability. So I'm not answering your question directly, but to answer your question directly, yes, we probably can. If we have the data, we can, these are quote unquote public data in a sense, with the exception of the ones that derive from patients directly.
So Tintin, if I could make a couple of comments or suggestions for you and James and Michaela and Patricia to sort of take back to the Cancer Center. So the first I would say is that it's fairly predictable that the NIH and other bodies in the not too distant future are actually going to require that not only do you know this information, but that you actually address it in your experimental designs, right? I mean, this is what happened a few years ago when there was all the problems with cell line misidentification. They had new stringency standards for doing STR profiling and so forth. Right. So I don't think this is a question of should we, I think we have to do it. Yeah. And I think the Cancer Center needs to take a leading role in promoting that. And the very simple thing to do, or I'm just throwing out examples, but would be for the executive committee to say, okay, for the next few years, half of every program budget is going to be dedicated to paying for this stuff, right? So you put a pot of money on the table and say, okay, everybody bring all your cell lines in. We're going to take it to Chris's core. We're going to give you as much ethnic information as we can. And obviously, if we do that for a few years, we're going to have an amazing database and resource that everybody can tap into. And again, I think it's inevitable that we will have to know that information. So we might as well get out in front of ahead of this. And I think, and I would be astonished if Rich wasn't, and the executive committee wasn't incredibly supportive of that. It might even put additional money on the table to do it. It shouldn't be just MCO. Obviously, it should be everybody in the Cancer Center. So maybe it should be a more centralized resource. But for some period of time, we should all be able to submit our lines and so forth to make it really easy. I will say, when the issue of cell line misidentification came up, there was a great deal of resistance in the scientific community. People didn't want to know that their cell line wasn't human, that they thought was human, or was prostate cancer turned out to be, something totally different. Yeah. So I think there will be resistance, but if we are sort of diligent and make these things as easily available as possible, then hopefully people will eventually come along.
Yeah, I'm thinking this is going to be like, sex as a variable, right? And yeah. It's now a required component of any experimental design. And so you're absolutely right. We are going to get to that point, and that we might as well get started on it.
Yeah, and the next CCSG renewal, it's going to be huge, right? So yeah. The more proactive we are, the better.
These should be data that are made available to everyone, right? I mean, one could, I don't know if it could be done easily, but one could take the cell line encyclopedia as a start, where they've sequenced just a ton of cell lines, and see if it could be, you could basically get ancestry information across all those cell lines, hopefully by some automatic method. So people don't have to submit their individual cell lines. First off, they could just go to a public resource and say, okay, is my cell line on this? And then what we could do is we could do cleanup and then find the cell lines that have not been done and add those to it, but probably add it to a national repository.
Chris, sorry, just real quick. Chris, what's the easiest way, if I have a particular cell line that the database says is particular ethnic group, but obviously I've been passaging it for years, I don't really know that. What's the methodologically, what's the easiest way to verify that? Are there polymorphisms that can be used to verify what I have is what it's supposed to be?
Yeah. Yeah, I mean, and then, we'd have to look a little bit at sensitivity if you're sort of passaging things and you start to get some, some dropout of certain chromosomes and stuff like that. But honestly, it should work pretty well. So yeah, and if you're thinking about sort of scoping out what a resource like this would be for a lot of cell lines, I mean, you're talking about costs. I don't know the exact costs, but maybe a couple $100 per sample at the most.
That's what we pay for STR profiling now, right? This is not a barrier.
So I think
and it would be a
just say-
one-time thing, right?
Yeah, you know,
compared to like
once and
STRs.
check, right?
Yeah.
Yeah, I was just going to say we probably have a lot of this information. My guess is that anytime we're talking about cell lines, the genotypes already exist. That is, if you're doing any functional genomics, the genotype information already exists. So in a sense, there's not a need to spend a lot of time doing something extra, as long as we can get access to that data, to those data for this purpose. And to echo what Craig, to add to what Craig was saying, I suppose you folks all know this, but if you're generating genomic data with NIH funds, you must submit that to the NIH as a public resource, right? My guess is that if we looked across the Cancer Center and asked who's actually doing that, the numbers are probably not spectacular as at most places. But that's also another nice way of making data, getting to a place where we're freeing the data, because this isn't just about the Cancer Center addressing underrepresented minorities and disparities. This is also about those slides that I was talking about, increasing the value of data. It's a fixed cost to produce it. The more we can get it out, the better off we all are. Again, it's a little bit aspirational, but if each of your groups, instead of saying, let's submit it to Chris, were to say, no, this year we will submit all our data, all our functional genomics data to this repository. Now you've captured all your data, you've put it all in one place, you've cataloged it. And now these kinds of questions that are really trivial to do from a data perspective, but really hard to do because of the socio-technical aspects of getting data in one place, cataloging it, defining what the questions are, those barriers go away if you can get to a place where I'm going to share my data and I'm going to share it in a way that allows other people to use it. So that's a soapbox here, but yeah.
Can I just bring this down to really basic understanding? So if Adam is saying like he has done studies on his DIPG cell lines that are new to him, like he's developed those, they have MAF numbers, they're not super common, but he's published on them and he's submitted some RNA-seq to GEO already, then you're saying we can just extract right now what the ethnicity is, right?
Yeah. So the way this would work is the SNPs, right? These are not associated. These are not cancer associated. SNPs, especially the common ones. All you need is a very small change in percentage and proportion from one population to the next in order to say that there's a difference. And that's why people use common SNPs, is that it's a small change that leads to a statistically significant difference. If you go for rare variants, it's harder to show that those are population specific because they're rare. So common SNPs are things that are going to show up in many of our functional genomics assays because they're common. And we don't need a large number of them, maybe 50 or 100. The larger the number, the more specific we can be in terms of assigning them to, instead of 2 or 3 ethnic groups or races, Chris's 127. But with a relatively small number, we can assign those to race or ethnicity. And that you can extract directly from RNA-seq data, ChIP-seq data, ATAC-seq data, even single cell data, which are challenging for a lot of other things to do. So
is it worse than funding a feasibility study to pay someone and go access, I'm just making this up, Adam's data that's publicly available, and do this analysis and get the ancestry information, and then once we have that, then try to expand it to, let's say, all MCO members' genomic data that's publicly available?
I mean, I'd start with where you have repositories already, like the Sanger's Online Encyclopedia, where
Well, no, that's all. I mean, that's part of our curation. Like that information's all out there. We have that. We're talking about things that are uniquely developed here, but are not primary patient samples necessarily. But cell lines, like Tintin has many.
Right. No, I was going to say that we could add those on, but has Sanger? I mean, can you go to Sanger? Have they already done this on all those cell lines?
Yeah,
I mean,
the CCLE has ancestry data.
Oh, okay. Well, that's
and
done. Yeah,
that's the kind of info we were going to make easily available.
Maybe the thing to have us the supported project would be somebody who writes the code so this can be done relatively easy with different forms of data. So, you know, someone could enter their ChIP-seq data, someone could enter their whole genome data, well, that might be easier, but other things like that. And it would, you know, basically provide that information, almost like a platform.
Mm-hmm. So is there someone like that that's currently available for hire, or is it how much work are we talking about? Are we talking about 1 FTE all the time, or is this like part-time work for somebody, or a postdoc that we can pay, you know, 25% time?
This genetic ancestry piece, or
like accessing available genomics and then coming up with ancestry information.
I mean, how many samples are, like, how many cell lines are we talking here? Maybe hundreds at the most, or is it?
Yeah.
Yeah, I mean, that's something we can probably help take care of on the CCPM end, since we're doing a lot of ancestry related work anyway. So
Chris,
we can kind of sidebar with Sean about the right way of passing data.
Chris and I can work on it. The challenge here is the processing is easy. It's getting the data organized and in the right place to do the analysis that's hard. The analysis will run on a laptop in a couple hours for any sample that you want to. And we could teach somebody to do it. But getting the data, getting access to the data, and having it in a form for analysis is the challenging part. So that's hard to write down.
Is geo submitted data not appropriate?
Yeah, with geo, I routinely mine geo for tens to hundreds of thousands, sometimes millions of samples. Doing that kind of thing is easy because the data are available. So
we have a lot
and for anyone who's
where that data is already there, they can just say, "Here's my GSE number." You know, go at it.
Yeah, and Chris and Sean, if you need funding to do that, if you need some FTE for somebody on it, just put in a request to the Cancer Center. I think this would be very well received.
Well, I mean, MCO was thinking about, we're willing to fund it too. So
okay, you can have it.
So I guess we need to figure out how many people, because there's a lot of people who, like me, who work on mice, and I know what the strain is already. You know, are there people interested in having that data mined? You know, I don't know how many people we're talking about in MCO.
What might be useful is for Chris and I to get together and define what the process looks like. And ideally, we would have this be at Colorado. I found that having a centralized process can be a real challenge to create, and sometimes isn't used in the way you'd like. So ideally, we'd have a somewhat distributed process. So we'd be able to write down what to do and the software to use and how to use it. And you can get results. And especially for folks like you know, James and Tintin, in Boulder, you guys have people who think about these kinds of things, so who are bioinformaticians. So if we were able to hand them a workflow to say, if you do this, you'll get data that's comparable to atoms, and we can compare apples to apples, that's a win. So I think Chris and I can talk about what that process looks like, and think about how easily we can write it down. And then maybe from there, we pilot with a couple of groups to work on their data, the ones that have well-organized and high-value data. And Adam, maybe yours is a group that we'd want to think through this with, and we can then go from there. And I think having a time window of, 4 years or so to think about this thoroughly is it's a nice time window to think through.
Great. Thanks a lot. I think we're hitting the end of the hour. So
Yeah, no, that was great discussion, I thought. Yeah. And Ellen, is there any way you can save the chat?
I already did it.
Oh, you did. Apparently I'm not allowed to for some reason, but yeah.
Oh.
Go.
I
Nice.
think it says saved.
Yeah, cool. Yeah, sounds good.
Patricia, if you can paste that into the Google Doc, that'd be awesome. So we can all just see it.
The whole chat.
Okay.
Yeah.
Yeah. And then we've got it all.
Okay.
Thanks for a great symposium, MCO people.
Yeah, this is really fun.
Thank you.
Thanks, guys. It's great. Yeah.
Great.
Great. Thanks,
Thank you,
everyone.
everyone. See you.
Thanks all.
Bye now.